Hello everyone. Again I am here with another lecture today, and today's topic is pentose phosphate pathway. After this we will talk about citric acid cycle or TCA cycle, and then we will look forward to complete this glucose metabolism. So today's topic I will start with the definition. So the pentose phosphate pathway, which is also known as phosphogluconate pathway and hexose monophosphate shunt, is a metabolic pathway parallel to glycolysis. It generates NADPH and pentose, which is 5-carbon sugar, as well as ribose 5-phosphate, a precursor for the synthesis of nucleotides. While the pentose phosphate pathway does involve oxidation of glucose, its primary role is anabolic rather than catabolic. The pathway is especially important in red blood cells, which is erythrocytes. Next, we will look into some important points about this pentose phosphate pathway before talking about the reactions. So first is rapidly dividing cells such as those of bone marrow, skin and intestinal mucosa, and those of tumors use pentose ribose 5-phosphate to make RNA, DNA and such coenzymes as ATP, NADH, FADH2 and coenzyme A. Next is tissues that carry out extensive fatty acid synthesis, which for example liver, adipose, lactating mammary gland, or very active synthesis of cholesterol and steroid hormone, which we can observe in liver gland gonads, require the NADPH provided by this pathway. Erythrocytes and the cells of the lens and the cornea are directly exposed to oxygen and thus to the damaging free radicals generated by oxygen. By maintaining or reducing atmosphere, a high ratio of NADPH to NADP+, and a high ratio of oxidative damage to protein, lipid and other sensitive molecules. So before starting the reaction, I would like to tell you that pentose phosphate pathway are having two phases. First phase is oxidative phase and next phase is non-oxidative phase. So first we will discuss about the oxidative phase of pentose phosphate pathway. So we will look into the first reaction. In the first reaction of pentose phosphate pathway, the oxidation of glucose 6-phosphate by glucose 6-phosphate dehydrogenase, which is also known as G6PD, to form, and it form 6-phosphoglucono-delta-lactone, which is an intramolecular ester, and it also needs NADP plus as the electron acceptor. So you can look into the reaction that glucose 6-phosphate is being converted into 6-phosphoglucono-lactone with the help of the enzyme glucose 6-phosphate dehydrogenase, and one NAD plus consumption is there, which is releasing NADPH. In the next step, the lactone is hydrolyzed to the free acid 6-phosphogluconate by a specific lactanase. Let's look into the reaction. 6-phosphogluconolactone is converted into 6-phosphogluconate with the help of the enzyme glucolactonase, where one water molecule consumption is there and which is generating H+. In the next reaction, the 6-phosphogluconate undergoes oxidation and decarboxylation by 6-phosphogluconate dehydrogenase to form a ketopentose, which is ribulose 5-phosphate. This reaction generates a second molecule of NADPH. This ribulose 5-phosphate is important in the regulation of glycolysis and gluconeogenesis. So we will look into the reaction now. So 6-phosphogluconate is being converted into ribulose 5-phosphate, which is a ketopentose, with the help of the enzyme 6-phosphogluconate dehydrogenase. And here one NADP plus molecule consumption is there, from where we are getting one molecule of NADPH and one molecule of carbon dioxide. And in the last reaction of oxidative phase, phosphopentose isomerase converts that ribulose 5-phosphate to the aldose isomer, which is ribose 5-phosphate. So this was the oxidative phase of pentose phosphate pathway, and the net result is the production of NADPH, a reductant for biosynthetic reactions, and ribose 5-phosphate, a precursor for nucleotide synthesis. So these NADPH and ribose 5-phosphates are the net result of this oxidative phase of pentose phosphate pathway. Next we will look into the non-oxidative phase of pentose phosphate pathway. So before going into the reaction, we will look into some important points about this. The non-oxidative phase links the glycolytic pathway to the pentose phosphate pathway, and it allows our cells a way to break down ribose molecules ingested into our body. This phase uses several important enzymes including phosphopentose isomerase, phosphopentose epimerase, transketolase, and transaldolase. In a series of rearrangements of the carbon skeletons, 6 5-carbon sugar phosphates are converted to 5 6-carbon sugar phosphates, completing the cycle and allowing continued oxidation of glucose 6-phosphate with production of NADPH. And continued recycling leads ultimately to the conversion of glucose 6-phosphate to 6-CO2. Two enzymes unique to the pentose phosphate pathway in this interconversions of sugars are transketolase and transaldolase, and transketolase catalyzes the transfer of a 2-carbon fragment to a ketose donor to an aldose donor. So now we will look into the reactions of non-oxidative phase of pentose phosphate pathway. In the first reaction, transketolase converts ribose 5-phosphate or xylose 5-phosphate to glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. So this is the first reaction, and in the next reaction, transaldolase catalyze a reaction similar to the aldose reaction of the glycolysis, which aldolase was being used and which was producing glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. So a 3-carbon fragment is removed from pseudoheptulose 7-phosphate and condensed with glyceraldehyde 3-phosphate, forming fructose 6-phosphate and glyceraldehyde, and the tetrose erythrose 3-phosphate. So if you look into the reaction, you can see glyceraldehyde 3-phosphate plus pseudoheptulase 7-phosphate, which was our end product of first reaction, that is being converted into fructose 6-phosphate plus erythrocyte 4-phosphate with the help of the enzyme transaldolase. In the third reaction, the transketolase, which requires a TPP in the both case. In the first reaction also transketolase requires TPP, and in the third reaction also transketolase need TPP for its action. As a cofactor acts again, forming fructose 6-phosphate and glyceraldehyde 3-phosphate from erythrose 4-phosphate and xylose 5-phosphate. And then those two end products can enter the glycolysis cycle again. So you can see in the reaction from fructose 6-phosphate, which can directly enter the glycolysis as it is an intermediate of glycolytic cycle, and the erythrose 4-phosphate, in order to enter the glycolysis, it need to be converted into some one of the intermediates glycolysis pathway. So with the help of the enzyme transketolase, both erythrose 4-phosphate and xylose 5-phosphate is converted into glyceraldehyde 3-phosphate and fructose 6-phosphate, which both are intermediates of glycolysis cycle, and they can enter into that. Next, we will talk about some important points about pentose phosphate pathway. So in the first phase of pentose phosphate pathway, it consists of two oxidation that convert glucose 6-phosphate to ribulose 5-phosphate and reduce NADP plus and NADPH. The second phase comprises non-oxidative steps that convert pentose phosphate to glucose 6-phosphate, which begins the cycle again. In the second phase, transketolase, which require TPP as a cofactor to work, and transaldolase catalyze the interconversion of 3, 4, 5, 6 and 7 carbon sugars with the reversible conversion of 6 pentose phosphates. In the carbon assimilating reactions of photosynthesis, the same enzyme catalyze the reverse process, the reductive pentose phosphate pathway. All the enzymes in the pentose phosphate pathway are located in the cytosol. A genetic defect in transketolase that lowers its affinity for the TPP cofactor exacerbates the Wernicke-Korsakoff syndrome. So about this syndrome I will talk about in another video, which will come under the genetic disorders. So this was pentose phosphate pathway. I hope I was able to make you understand about pentose phosphate pathway in a very detailed way. If you like my video, don't forget to like, comment and subscribe and share it with your friends. And I'm also available in Instagram. Here is my Instagram ID. And also in Facebook, I have a page where I post about job updates regarding this biotechnology and microbiology field mainly. So you can follow my page there. And if you are having any query, my mail ID is also given in the description box. You can mail me. I hope to get back to you soon with another video.